here today I am going to teach you about the third line of defense system. So it basically the one main point is that it gives cellular and molecular response. Okay, so here it is divided into the two parts. The third line of system is divided into the two parts. Number one is cell mediated response. While number second one is humoral response. Okay, so here you can see, so here first of all, the cells which are present in the cell mediated, these just kill the pathogen or the antigen. So these are T cells or the T lymphocytes. T cells or T lymphocytes, and the second one is macrophages, while third one is dendrite cells. Okay, so talking about the humoral, it has basically the B cells or the B lymphocyte. Those are divided into the two parts again. One is plasma cell, and other is memory cell. It is invisible. Okay, so here I am going to discuss about the T lymphocytes. Those are basically present in the stem cells, like in the bone marrow. But and during the maturation, they are done in the thymus. The maturation is in thymus. Okay, so talking about the B cell, so let me do like this. So the B cells are, their maturation is done in the bone marrow. But there is a specific region in the bird that is bursa of birds. Bursa of birds. Okay, but here we have that one assumption that in the animal cell, like here I am writing, in the human cell, it is mostly in the spleen. Spleen is the organ, and though that is in the lymph node, or the lymph node is known as a checkpoint too. Okay, so now moving towards another, like as I told you, these here we have the cell mediated, they just directly kill the cell. Not all the cells, but kill the specific one cell. Okay, so here I want to tell you a little bit on the antigen. What is antigen? Okay, so let me write here. Antigen is basically, okay, so antigen is any foreign particle, or you can say pathogen. Any foreign particle, or you can say pathogen. Okay, which trigger immune system. Which trigger immune response. मीन्स एंटीजन एक ऐसा सब्सटेंस नहीं, एक ऐसा सेल या बैक्टीरिया कह सकते हैं जो कि हमारे इम्यून सिस्टम के ऊपर ट्रिगर करता है। कोई भी एक फॉरेन पार्टिकल या कोई पैथोजन हमारी बॉडी के अंदर इंटर हो गया है तो मीन उसके ज़रिए हम मालूम कर सकते हैं, मीन्स ये एक तरह का ट्रिगर करता है हमारे इम्यून सिस्टम के एंटर हो चुका है कोई पैथोजन। ओके, तो अभी हम डिस्कस करते हैं सेल मेडिएटेड रिस्पॉन्स। So let's see here. So here we have cell mediated immune system. Okay, so here the major cell play the main role. Those are role of monocytes. And these converge into the macrophages, like the conversion of monocytes into the macrophages. Okay, so here these are basically, let me tell you. Yeah, so here the monocytes have the one major, you can say, think like these can 1 to 20 hours these can stay in the blood. So this is the one most important thing, like monocytes can stay 10 to 20 hours in the blood. So again then they will move to the intracellular spaces of macrophages. Move to intracellular spaces of macrophages. Okay, why? Because of the, I'm writing here, because of the growth and increase the size. Okay, for the growth and the size increasement. So now what are macrophages? Here I am going to explain you. Here macrophages are the big cells which eat which work as the engulfing. Just they eat the phagocytes or phagocytosis. Just do the phagocyte and phagocytosis of any pathogen. Okay, so here we are talking about the MHC1 and MHC2. So MHC play the main role in the, we can say, macrophages, that role of macrophages. Here you can see, if here we have any one cell, so here the antigen came. This is the antigen. Antigen come inside, then this phagocyte, what the macrophages did, it did the work of engulfing, means just direct eat this, otherwise it break down these into the small pieces. After doing that, it represent these pieces on the surface. How they represent? With the help of MHC1. They represent with the MHC1, and these MHC are the protein nucleated cells. What these are? Protein nucleated cells. Okay, so here they just like this. These are the MHC1 and they represent the antigen here. So these now are known as anti-presenting cells. APS means anti-presenting cells. So what is the full form of MHC? This is the major histocompatibility. Major histocompatibility. Okay, so here we have like one more in the human, that is HLA. This is known as human lymphocyte antigen, which is present in the humans. Okay, so hopefully that is clear to you till that. So now I am going to give you concept on the MSc1 and MSc2. So let's discuss. Okay, so here we have here we have the structure of MSc1 like this. Here we have the alpha 1, alpha 2, alpha 3 and beta 1, B1. Why here it is the beta 1? Because of here we have 3 alpha and 1 beta. Okay, so now these are combined with the polypeptide bond with the subunit, that is alpha and beta. Here these all have the tails too. Okay, so these are combined with polypeptide bond okay, so these all combine like antigen combined with subunit, that is alpha 1 and alpha 2. Here these will combine on the alpha 1 and alpha 2. Okay, so these are the amino acid sequences. What amino acid sequence? Okay, so hopefully till that is clear. Now moving towards another point, like here MSc1 always have CD4 receptor. Okay, CD4 receptor, and they have the cytotoxic T cells and the natural killer cells, and here the macrophages release okay, so this is made up of 8 to 10 amino acid chain. Okay, so now moving towards let me tell you one more point here. Okay, so MSc1 is present in the all nucleated cells, like here I am writing all nucleated cells except in the nerve cells and platelets. Okay, so the size of this is 45 kd, means kilo Dalton, and 1 Dalton is equal to 1 AMU. Okay, so here let me make this. Yeah, okay, it's done. Now moving towards let me tell you here why we put the B1. It is also known as BM. Okay, so their full form is microglobulin which have no membrane. That's why this is different from these three, and here these are covered with the one type of membrane, but this one is not covered with the membrane. So this was all about the MSc1. Now moving towards MSc2. So here you can see here the cells are present. Those are macrophages. And dendrite cells. One more cells by B cells. Okay. So they all work as the engulfing, means they just directly eat the bacteria pathogen or any antigen, they directly eat like that. Engulf the foreign engulf the, you can say here, foreign particle, foreign antigen. Alright, so here the T helper cells are used. Why T helper cells? Because they release one type of, we can say, toxic material, that is, they release the interleukin 2, I guess. And confirm it again, interleukin 2, that is very toxic. Okay, and here we have CD. Oh sorry, here we was having CD8 receptor. Here we have CD4 receptor, and we check this answer like this. Okay, so this contains the two peptide chains, consist of two polypeptide chain, alpha and beta chain. Okay, so now here the one most important thing is, here the size of MSC1 was 45 kilodalton, while here the size is, like here we have the two chains different, alpha chain is different, beta chain is different, so the both have different size. The size alpha is 33 kilodalton, okay, and the beta have 28 kilodalton. Okay, so their structure is basically like this. They are connected with, we can say, they are connected with the cytoplasmic cell here. And it has two chains, one is alpha and one is beta. Okay, so here you can see these both are covered with the membrane. Why they are working? These both are covered with the membrane. Here you can see. Okay, so here the any antigen comes, so they will represent the antigen on this one side. Like this is the alpha 1, this is the beta 1, this is alpha 2 and beta 2. So they will represent over here on the alpha 1 and beta 1. Hopefully it's clear all about MSC1 and MSC2. Bye bye. Again we will continue this from tomorrow. Okay, Allah Hafiz. In next video we will continue this. Bye bye.